The second part of this topic is basically we'll start off with the application of recombinant DNA technology and basically how you could do it theoretically. So first and foremost, okay, if you were to analyze gene structure and expression, so basically you could do some recombinant DNA and then utilize the technology, use suitable expression vector. Secondly, for pharmaceutical products, they are usually made using recombinant technology, whether you want to produce drugs or vaccines like what we're looking for COVID-19 nowadays. The third is for the GMO or genetically modified organisms, whether transgenic plants or transgenic animals. And of course, the application in medicine and a whole lot more in the food industry, in the textile, in our everyday life. Okay, so a lot of it could be applied the recombinant DNA technology. So uh, as an example, so these are some of the protein products of recombinant DNA technology. So well, we have human insulin here, okay, which is very famous. Okay, so it has been firstly made in E. coli, and of course, treatment is for diabetes and a whole lot other more. And as you can see, okay, all of them here, the product are from eukaryote human cell, okay, and they are made in E. coli, okay. Saccharomyces cerevisiae, okay, mammalian cells. This is eukaryote as well, okay. But this, again, all of them, okay, is basically in bacteria. So most of the time, this is not uncommon. This is quite kind of like a normal norm. So you use recombinant cells to mass produce proteins, okay, from either using bacteria or yeast or mammalian. And I think somewhere along these slides, towards the end of the second video, okay, we will have a look at the differences and the pro and cons, okay, of having or basically of using these three in the comparison as an expression vector. So selection of protein production host is actually very crucial. So it would depend on, I think, about 4 to 5 factors. Okay, the first is the protein to be produced. So whether the protein comes from prokaryote or eukaryote, because it does matter. Okay, so secondly, okay, whether it's cytoplasmic or secreted. Okay, again, this is also crucial. Okay, because if it's in cytoplasmic, okay, usually there's not really that much of a problem. But if it is secreted, you have to choose a suitable host that could basically allow the secretion of the product later on. Okay, and lastly is the organelle specific. Is it organelle specific or not? Especially if the protein comes from eukaryotic cells, okay, for example, plant or animal. So probably they have a specific organelle. So you need to also choose, okay, a host that would enable it to be expressed, okay, in that specific organelle. Secondly is the amount needed of the protein, okay, to be produced. So whether it's for analytical, okay, which is only in nanogram, functional studies also not that much nanogram, antibody production, you may need quite a lot, okay, because you may want to test it out. Oh, sorry, of the spelling. Structural biology, okay, this is basically up until when you want to proceed for the X-ray diffraction, and of course for commercialization, okay, you need lots and lots of them up until kilogram. Now, the third one is the demands of authenticity. Okay, so whether it should be active or inactive, okay, or whether it should be native or denatured, okay, the form of the protein. Now, before you say that denatured protein is not good, no, okay, it depends. Not all protein are denatured at high temperature or when mixed, okay, with certain chemicals. Sometimes certain protein, okay, when they are not in their full structure, that's where they are best served or where they would function the best. Secondly is the modified or unmodified version of the protein, okay, whether it goes through PTM or not, post-translational modification. The third one is molecular weight, okay, the size of it, okay. The fourth one is the sequence, okay, so you need to actually know, okay, the sequence, whether it's going to have similar exact sequence or you're going to modify the sequence, okay, do a bit of metagenesis and whatnot. And of course, okay, the last is actually on the expression. So the fourth is the cost, okay. Probably you're wondering why the cost is not the first. Yes, the cost comes into view, okay, but you also need to look at the other three first, okay, because you need to choose on that one first before determining the cost. And lastly is the protein production. So not to forget, okay, you have to look at the systems available in the lab, okay, in your lab or the facilities available, okay, nearest to you or the one that you could use. Now, how do you express your protein? So you need to look at these three elements: the host, the cofactors, and also the chaperone or second protein for correct folding. So Why is the host important? We have seen, okay, in the first video and in earlier part, okay, of this video, that the host is actually important, okay, if you were to express your protein correctly. So for example, okay, mammalian cells, okay, usually it's the slowest and the most expensive, but it has been deemed as the best for human proteins. For insect cells or animal cells, any other animals apart from mammalian cells, they have similar advantage to mammalian cells, okay, which means that they are faster and also cheaper. Okay, but of course they are actually quite cheaper as compared to mammalian cells. The third one would be the yeast expression system. It is usually an option to bacteria if bacteria is basically not a success for you, but usually would affect protein, okay, with PTM. And extraction is usually not always easy with yeast. Usually people would try not to use yeast unless they have no other option. Okay, if you were to choose, okay, between yeast and mammalian cells, of course people would choose yeast. But if you were to choose between yeast and E. coli, people would choose E. coli. And the last one would be the E. coli, because it is of course the cheapest, easiest, and the fastest. But okay, everything has a but. Okay, it is not suitable to all protein, especially for eukaryotic gene from higher animals, okay, such as us humans sometimes. So second element is the cofactors, okay. Are there cofactors needed to express your protein? Okay, so that's why you need to study the protein, okay, intensely in detail. Know what does it need cofactors, or it could function without the cofactors. If there is no cofactor needed, that is fine. But if there is a cofactor needed, cofactor or cofactors, then you have to take into account of how you can get your protein, okay, to be expressed in the host. Okay, and the last one, okay, would be whether it needs a chaperone or second protein for correct folding. Now we know that protein, okay, could only function when it is folded correctly in its tertiary up until the quaternary structure. So sometimes, okay, it basically needs, okay, a second protein to help with that, because usually protein works doesn't really work alone. Okay, usually they work, okay, in group, okay, whether a couple, okay, or basically 2-3 of them, okay, together. Or sometimes they need a protein called a chaperone protein for correct folding of the protein to deliver its biological function. Now, let's have a look, okay, at the characteristics of the 4 different expression systems. We have seen E. coli, yeast, mammalian, and insect just now. So insect is other options, okay, apart from mammalian. Both of them basically are for animal or from animal. Now you don't see anything from plant, right? So why is there anything not from plant? Okay, so give it a thought, okay, and if we have time, probably we will discuss on that. So these are some of the characteristics, okay, so you can do a comparison over here. I'm not going to go through, okay, all of them in details, okay, so you can pour on this, okay, look at your notes, okay, and try to understand how they are different, how they could benefit. So usually these characteristics would be crucial in your work if you were to express your gene in an expression system, and you may want to take a look, okay, at the 5 factors, okay, that we have discussed earlier, okay, of how to ensure that your protein could be expressed and in a good way. Now let's have a look and also a comparison, okay, between prokaryotic and also eukaryotic host system. So for prokaryotic system, okay, we're going to take a look at E. coli, of course, okay, the most famous. Apart from E. coli, there are other systems, okay, from Bacillus, but usually people prefer E. coli, and also Saccharomyces. All right, so the advantage, okay, is as such, there are many references, and of course, okay, a lot of experience available, because E. coli is the most common, and it is basically the kind of like the main bacteria to be used. So apart from that, there are also wide choice of cloning vectors, so gene expression is easily controlled, and it is also fairly easy to grow with high yield, and the product can be designed for secretion into the growth media. But okay, everything has its pros and cons. So the cons or the disadvantages are no post-translational modification, because, well, it's no-brainer, because this is a prokaryotic we're talking about. Biological activity and immunogenicity may differ from the natural protein, since it basically couldn't do PTM. And this has been monitored quite a lot, okay, for eukaryotic cells, okay, high endotoxin, okay, content in the gram negative, okay, and we know what kind E. coli is. So for the eukaryotic systems, okay, we're going to take a look at yeast, okay. So the advantage is that it lacks detectable endotoxins, okay. So fermentation is relatively inexpensive if you know how to tweak it. So facilitates glycosylation and formation of disulfide bonds. So glycosylation is one form of PTM or post-translational modification, and formation of disulfide bond is actually quite crucial for protein to be compact. So only 0.5% of native proteins are secreted, so isolation of secreted product is simplified, which means that it doesn't really deviate that much, or there would be no mutation, or not say no mutation, not higher mutation occurrence. Okay, and it is well established, okay, in large scale production and downstream processing. So the downside, okay, is that gene expression is less easily controlled, okay, because yeast has a mind of its own, even though it's eukaryote, okay, but because it has a different life cycle, okay. And secondly, okay, even though it facilitates glycosylation usually is not identical to mammalian systems. So if anything is from the mammalian cells, okay, if you were to use this, you have to be careful, especially if you know that your protein goes through PTM and the PTM is in the form of glycosylation. Now, this is basically just a general workflow of the recombinant of the fusion protein. So you may see that probably it is a bit different from the recombinant DNA, but more or less they are quite similar. It's just that because you are using, okay, a protein, you are ensuring, okay, in the beginning that, okay, you will get the protein in the end, okay, to be isolated or purified later on. So you can choose your protein expression cell line, whether it's from E. coli or insect and mammalian or from yeast. Okay, choose your expression vector or system. So insert DNA coding region into the plasmid with restriction enzymes to facilitate the ligation later on. So you grow the cells in minimal medium. So there are varieties of minimal medium depending on the kind that you use. Okay, and also it actually depends, okay, on the expression system. So the labeling strategy, okay, so probably it's not, it's quite blurry because I basically stretch this diagram. Basically it depends, okay, from one protein to another. So you can use a general protein label, you can use a commercial protein label, you can use radioactive, but of course, again, nowadays we don't really use the radioactive. So basically whatever that is available to you, you can just use it. So there are tons of labeling strategies, so you may need to have a read and see which would serve you better. So once you have done your labeling, this is to make sure, okay, that you could identify your protein later on. You could harvest the cells, and later on you need to lyse the cells, okay, to make sure that you could purify your protein. So purification of protein over here, the last part, is basically a bit tricky and not always easy. Why? Because it has to be done through chromatography. So you need to choose the best and the correct chromatography for your protein. So we have a few chromatography, okay, size exclusion, ion exchange, affinity, and also reverse phase. So especially if you are using ion exchange, make sure whether you are using anion or cation exchange. You need to be very careful about that because the buffer, the elution buffer to be used also matters in any one of this chromatography. And sometimes, I wouldn't say sometimes, in a lot of times, you may need to use two chromatography, okay, to make sure that you really get your purified protein. So among them, okay, usually ion size exclusion is quite common because that is just based on the size. But okay, ion exchange, size exclusion and ion exchange, okay, used as a couple usually is quite common, and or size exclusion with affinity, or basically reverse phase with size exclusion, because usually they are eliminated by size, okay, then only, okay, they are specifically purified, okay, whether based on affinity or basically based on the charge, and also basically whether they are polar or non-polar. Now let's have a look general outlook of the features of E. coli as the expression host. So the protein expression recovery sites in E. coli, okay, is whether it is through the cytoplasm, okay, basically over here, periplasmic, or via medium, which means that when you basically just grow it, okay, in the lab, okay, it doesn't really matter, okay, whether it needs to be in any part of it or you don't really have a target, so it's fine. Okay, so why basically this is important, okay, in the next slide, okay, because there are advantages and disadvantages in the protein production into each compartment. So you can just read on from your notes, okay, what are the advantages and disadvantages, because I'm not going to say that if you have something expressed in periplasm is better as compared to cytoplasm and vice versa, because it basically depends. Because sometimes certain protein are being secreted in the periplasm, sometimes in the cytoplasm, sometimes it is extracellular from cytoplasm to the extracellular matrix. So it basically have its own advantage and disadvantages, but you need to know how the protein is going to be expressed. You cannot have a zero knowledge on this. So now, which combination for success? So I've put that in the first point that there is no perfect combination, okay, because it's impossible. We would try most of the time, okay, our best, okay, we try our best, okay, to have a good combination, but it is almost impossible to get a good combination. You have to do lots of trial and error. You have to tweak here and there. You have to do lots of troubleshooting, I think, in the next slide. So multiple conditions usually should be tested. First and foremost, how many constructs, okay, do you need to be expressed, okay, the gene or the protein, because it really matters. Do you have only one gene or one protein, and basically how much of it, okay, do you want to be expressed? Is it abundant, okay, is it low or high abundance, okay, or whether the gene copy number and so forth. How many expression vectors, okay, do you need to use, okay, are they from the same or from different? Same meaning that if your gene or protein is from prokaryote, is it going to be prokaryote as well, or are you going to switch to eukaryote? Okay, so you need to know this. And how many hosts to be transformed into. Now, you can actually use more than one host, okay, to transform, similar with vector, okay. So it depends on your work, because when one vector is not suitable or there is something wrong with the gene, usually we shift another vector or another host. Now, okay, alright, so this is the troubleshooting slide. So troubleshooting usually has to be done in a lot of molecular work, okay, simply said. So if there is no or low gene expression or protein production, what you need to look at is look back at your promoter or vector or the host, okay, because, okay, chances are, okay, any one of them or all of them, okay, may be unsuitable. So you need to look at the structure of the promoter vector and the host. You need to look at the multiple cloning site. You need to look at the origin of replication, and you also need to look at how it is being expressed, or how the gene, when it is incorporated in, how it will be expressed. Secondly, low copy number gene usually needs high copy number vector and specific promoter. So this is where it comes back, okay, to this, okay, how many constructs, okay, do you need? So if you know that your gene, okay, if you're basically expressing a gene, okay, if it's a low copy number, usually you need a high copy number vector that could multiply it or could increase its expression, and usually specific promoter is needed for that, which means that you may need to design your own promoter. So the third one is the protein toxicity. If you are using, or if you are basically constructing a fusion protein, so protein toxicity prior to or after the induction, which is quite often occurring. And the last one is the codon bias, especially for protein. So, okay, as you can see, okay, I have this. Well, if you were to print it in black and white, you wouldn't be able, but in your notes, if you were to view it in your laptop and over here in the video, you can see that, okay, some of them are in, not say some, one of the point is actually in black, again, one of the point is in green, two other is in blue, okay. So this is for the general, okay, generally affect anything, okay. This is when basically it has to do with gene, and these are when it has to do with protein, okay. So another thing, again, that needs a lot of troubleshooting, especially in protein work, is the protein inactivity, okay, because protein is not always easy to work with. That's why not many people actually like to work with protein. So if there is protein inactivity, okay, most of the time, okay, it is caused by incomplete folding, okay. So when the folding is incomplete, the protein couldn't serve its best role or function, and sometimes, okay, there is also a CDNA mutation occurring, okay, that basically result in the protein to be inactive. Okay, so the last part of this video is actually on the summary of the expression system. So you can see over here, okay, is the mammalian, insect, yeast, and also bacteria from low to high, okay. In terms of the speed as in when you use it, okay, how long can you get your gene or protein to be expressed, okay. Of course, the lowest would be, or the speed, the I won't say the lowest speed, okay, the one that will take up the most time, okay, would be mammalian, okay, and the fastest, okay, would be bacteria. In terms of the cost, because we have to look at all of this, the cost, okay, the cheapest, of course, would be bacteria. The most expensive would be mammalian. Typical yield, which means that the production, okay, of the gene or protein expression, usually, okay, low would be in mammalian, okay, and the highest would be in yeast. That's why a lot of time, okay, people like to use yeast. Okay, why not bacteria? PTM. Okay, so number 4, okay, that's why we come to number 4, PTM, post-translational modification. So usually bacteria, okay, would be the lowest point, okay, because it doesn't have the mechanism to allow for PTM. So if you know that your protein occurs or basically goes through PTM, if you use the bacteria, chances are you may not get your your protein to be expressed properly or correctly, or probably it would be inactive. Okay, so the highest one would be in mammalian. Alright, so ponder on this, okay, and then move on to the third and the last video.